All right, in the last section, we talked a lot about the vocabularies associated with these things and kind of what's going on a general basis. So now in this section, what we're going to do is we're going to look at and see how this whole thing fits in with mitosis. And what we need to kind of keep in mind is that the cell cycle was this carefully coordinated sequence of events that gives rise to two genetically identical daughter cells, two clones of that original cell. And when we have errors in that process somewhere along the way or in multiple spots along the way, that's what's going to give rise to a potential tumor cell. Okay, so in its simplest form, what cancer is runaway cell division. In other words, the cell cycle is out of control. It's not being regulated. It's not being controlled. These errors aren't being sensed, or any combination of those different types of things. And if these things go unchecked, this is what's going to give rise to this rogue cell that we call a tumor. And it has, a faulty cell cycle, and that's what's going to give rise to, cancer cells, metastasis, and so on and so forth. Okay. So we talked about the P53 gene, and there's a lot of different types of genes that behave, in a similar fashion, but P53 is the really important one. And these genes contain certain types of components that act to suppress the tumor that is potentially growing. And what P53 gene does is it codes for transcription factors, and these types of proteins are going to promote the synthesis of these cell inhibiting or cell cycle inhibiting proteins. In other words, the P53 gene is going to make these things that's going to stop the cell cycle so that these errors can be repaired. Okay, so some tumor suppressor proteins from P53 can repair the damaged DNA. Others control the adhesion of the cells to one another. That control this idea of this density-dependent inhibition that we were talking about. And yet, other types of these components can have an effect on the cell cycle pathway, the signaling pathway that occurs. So P53 does a number of different types of things. And what normal P53 does is it acts to control the tumor formation using two main mechanisms. It's going to activate P21, which we talked about in the last section, in response to any damage or DNA. So when P53 is going to sense some damage to the DNA, it's going to activate P21, and that's going to have some responsibility in repairing the DNA that we'll discuss here shortly. And it also controls tumor formation by inducing apoptosis. Okay, so P53 expression activates P21. P21 is another gene that produces a protein that's going to halt the cell cycle in G1 and S, and it's going to allow these different types of cyclin-dependent kinases to become activated that are needed during this, they're going to become inhibited that are needed in the normal S phase that we talked about in one of the earlier sections here. What the S phase is doing is it's synthesizing the DNA. There's a lot of cyclin-dependent kinases that are necessary in order to synthesize the DNA. But what the P21 protein is going to do is it's going to bind to some of these CDKs, and it's going to prevent the S phase from occurring. Okay, and this is the type of halt step that's needed so that the cell can actually repair its DNA. Under normal conditions, what happens is that P53 gene activates this inhibitor gene when the DNA becomes damaged. And that P21 again arrests the cell cycle and prevents this cell proliferation, the division of a faulty cell, because we don't want a damaged cell dividing, because that's what's going to give rise to the tumor cell or the cancer. And if the P53 function is lost, then you can see what happens if we have a mutation in P53, we don't turn on P21, and if P21 doesn't get turned on, then we don't have any P21 protein available to stop the signal and to prevent the damage from being propagated into the new cells. Okay, we don't have any stop signal for the cell division, as it says here, and you can see that now the cell cycle is going to continue despite the fact that there's damage to the DNA. So that's why P53 is important, because it senses these things, and when it becomes damaged, you don't have any way to really sense these errors that are going on. And then as a result of this, you get an increase in these mutant cell proliferation and then the formation of a tumor. Okay, so this video will be something that will be posted, and you guys can watch this one as well. Okay, so now the P53, in addition to some of the other things that we've just described, it acts as an activator to express a bunch of these little what are called microRNAs. They're these, we said that we have transfer RNA, we have ribosomal RNA, we have messenger RNA. There's a whole bunch of different types of RNAs. And this miRNA or microRNA is another example of an RNA molecule. And these microRNAs get activated by P53, and they work to inhibit the cell cycle. And what they're doing is they're directly turning on genes that are involved in DNA repair just by regulating kind of the expression of these genes. And once this DNA is been determined to be damaged beyond repair, what these microRNAs and this P53 can do is they can stimulate apoptosis so that we can get rid of that cell and allow the immune system to kind of scavenge it up. Okay, and what we do again here, just the induction of apoptosis, the normal P53 gene, when it's functioning, can induce this cell death, this apoptosis, so that we can prevent this cell from proliferating. And again, the immune system comes and scavenges it up. And one way you can look at kind of the activation of P53 when it's working normally is if we go out into the sun and we get a sunburn from this UV light. The UV light, what it's actually doing is it's damaging the DNA in your skin. And what that does is it triggers this signaling pathway through P53 that blocks that cell cycle until the damaged DNA can get repaired. And if we can't repair that damaged DNA, you guys know that the cell goes through apoptosis or programmed cell death. And what that really is if you get burned on your skin from the UV radiation from being in the sun and your skin peels, those are dead cells that are peeling away because this P53 gene has done its actual job and it's induced apoptosis because it's been determined that the UV light has damaged the skin cells beyond repair, so it gets rid of those so that you don't propagate those damaged things that could form potentially a tumor. Okay, if P53 doesn't act that way, again, you can get these chromosomal abnormalities being perpetuated, proliferating, and giving rise to a tumor. Okay, so you can see here how P53 acts as a gatekeeper, and it's involved in a number of different types of pathways that prevent the formation of these mutations and the passing on of those things. Okay, another group of proto-oncogenes that you find in your cells. Again, these are genes that if they get mutated become oncogenes and can give rise to tumors, but this belongs to the RAS family of genes, and this the name comes from the studying these things in rats. They're called rat sarcoma, and these give rise to tumors in rat cells. So when they were studying these things, that's where the name of this protein came from. And when you get a mutation in a RAS gene, it can become an oncogene and possibly lead to the result or the formation of a tumor as a result of that. Okay, so these RAS genes, what they do is they code for these G proteins, and these G proteins are signals that promote really the growth and division of a cell. Okay, and they relay these signals from certain growth factors that kind of give rise to a signal cascade. In other words, it's a series of events that occur that give rise to the synthesis of proteins and the stimulation of the actual cell cycle itself. And under normal conditions, what this pathway does is it just operates according to the normal signals that it receives. It's not going to work unless it gets the appropriate growth factor. But if you get a mutation in the RAS gene and it leads to kind of this hyperactive RAS protein that signals this cascade of events that results in the cell growth and division, in the absence of this growth factor, then it's turning on genes that can be involved in cell growth, cell differentiation, cell division when it's not necessary, and that's what could potentially give rise to a tumor. Okay, if these signals are the result of cell growth and mitosis, if you have an overactive RAS signaling pathway, this is what you can see leads to a particular type of a cancer or a sarcoma in the case of these rats. Okay, so the takeaway here, the main thing to kind of take away from this is that there's a number of different types of genes that have been shown to play a role in caring for the genome and suppressing tumor formation. And if you have any kind of error that's occurring in any of these types of genes, it can lead to the formation of a tumor or cancer or something like that. And studying these genes and their proteins has better enabled people to really understand how the cell cycle works and really how to treat various ailments, cancers, tumors, etc. when they arise by knowing what's going on in these different types of pathways that occur. Okay, so make sure that you take any questions that you have and write them down. We'll talk about them in class. I know there's a lot here. It can be kind of complicated, but it's very easy to kind of answer these questions and gain a better understanding and more perspective from what we've just talked about here, just trying to kind of broaden your background information a little bit. And we'll go through the understandings in class. We'll answer the questions, and we'll make sense of all this. Okay, so we'll talk to you soon.